Welcome back, and here we go into our second unit of biomolecules. So looking at how carbon atoms can be linked together to form these backbones that we can then add functional groups that are going to give us the molecules of life. So carbohydrates, proteins, lipids, and nucleic acids. So we're going to be looking at the building blocks of life here, and we're going to continue our discussion of carbon chemistry into how do we link these pieces together. So the big thing that we have to learn about this unit is I'm going to introduce two terms. So the first term is going to be the word monomer. And as you may recognize, the prefix mono means one. So we're going to be taking these units of 1 units, these monomers, and we're going to learn how do we link them together to make polymers. So the polymers are what we know as the biomolecules when we talk about carbohydrates or saturated fats. Those are polymers. And you may also know the prefix poly means many. So we're just looking at taking 1 unit and linking them together into a chain of many units. So if you look at the diagram here, so building blocks of the cells, we've got sugars or carbohydrates, and when we link those together, we get polysaccharides. We have fatty acids, which can be linked together to give us fats and lipids. We have amino acids, that's the monomers that can be linked together to make proteins, and then we've got nucleotides that are the monomer that can be linked together to make nucleic acids. So here's a quick diagram that shows the structure of monomers and polymers. So a monomer is a small molecule. I would add to that a single unit. Okay? And a polymer is just a long chain molecule made up of repeated patterns of monomers. So if we look here, if we take our monomer from the top diagram, we can see that there are 1, 2, 3, 4 monomers that have been linked together to make the polymer in the picture here. Here's another diagram of it. So the monomers and then a process of polymerization will link them together to make polymers. And this is what we're going to look at. So these bonds in between here, how do we make those and make this polymer? How do we grow the chain for that? And that's what we're going to concentrate on in the next couple slides. So how do we go from small individual pieces to a large chain? And that process that we use is called a dehydration or condensation reaction. You need to be able to recognize both of those terms. They mean the same thing, but when something is dehydrated, what do? You're taking water out of it. Okay? And that's exactly what happens when we link monomers into polymers. So in the top line of the diagram here is an unlinked monomer. So there's one. And over to the left, we've got a short polymer. So we've already got 1, 2, 3 monomers that have been linked together. If we want to add a fourth, you will see there are functional groups. So look at this group right here on the unlinked monomer. Do you recognize what the OH functional group is? It is the hydroxyl group. And OH and this one right here, there's an H sticking off. And if we put those two together, they will form a covalent bond, and H2O, water, will be removed. That's why it's called a dehydration reaction. And the result is that we have just formed a new bond between monomers 3 and 4, and our chain has grown longer. Okay? So the condensation or dehydration reaction is critical to understanding how we put these monomers together. So here's a real example, and we haven't looked at amino acids yet, but on the very top line here, there are 2 amino acids. And what's going to happen is we're going to link them together by a condensation reaction, the removal of water. So just like we saw before, here is our hydroxyl group. Here is a lone hydrogen. And when those are brought next to each other, we can remove H2O. So water comes out, And in this case, between amino acids, we're starting a polypeptide. So this is a peptide bond. So between the carbon and nitrogen. So water has been removed, and we link those together, and that can continue hundreds of thousands of times as we build our polypeptide chain. So there's a specific example of a condensation or dehydration reaction. Now, what about the opposite? What if we have a long chain, in this case here, 4 monomers linked together already? What if we want to break it down? So the opposite of a condensation or dehydration reaction is hydrolysis. And if we look at it, hydro means water, and lysis means to split. So hydrolysis literally means the splitting of water. And if we want to break up the polymer, if we took water out before, we've got to add water back in. So if we see right here, we're going to add water back in. That water gets split. It's going to break the bond between monomers 3 and 4, and look what happens. We get a hydroxyl group on one end, a hydrogen on the other, and we have reversed the process. So hydrolysis adds a water molecule to break that bond. Okay, so there's the opposite. So now we know a little bit of how we can put molecules together, how we can take them apart, and this will help in our understanding of the biomolecules. So here's a real example of sucrose. So this is table sugar, and they are linked together right here. There's an oxygen between them, and when we digest sucrose, we've got to get it down to what's called a monosaccharide, down to glucose that our body can use. So what ends up happening is we add water, and where that single oxygen was right there, there's that original oxygen. We add a hydrogen to that and an OH to the other, so we've got our H2O, and now we've split it into 2 monosaccharides of glucose and fructose. So there's an example how we use it to split molecules apart. Okay, and that leads us right to the biomolecules. So carbon compounds include carbohydrates, lipids, nucleic acids, and proteins. I'm not going to go through this whole table. It's in the media album at the beginning of the unit, so you should look at that. But it's going to show basically the elements that it contains and the monomers that it forms from. And finally, I'm going to show you this. I know it's a lot to look at right now, and I'm not going to go through the whole thing, but I just want you to know that this chart is available in the media album, and this is a great one to look at in terms of wrapping up the thing, in terms of what the function of the different biomolecules are. But what I want to do right now is just look at 1 through 6 here, the basics. So number 1, carbon is the central atom. So we talked about how it has 4 valence electrons. Carbon will bond covalently because of those 4 valence electrons. Carbon likes to share electrons with sulfur, phosphorus, oxygen, nitrogen, and other carbon and hydrogens. Sponge. Okay, now we've talked about oxygen, nitrogen, carbon, and hydrogen as being the big 4 elements that make up living things, and the SP are going to be sulfur and phosphorus. And because of the backbone of nucleotides and the structure of proteins, we add in those 2 elements as well when we talk about biomolecules. All 4 groups, so carbohydrates, proteins, nucleic acid, and lipids, are big molecules. They're macromolecules. And finally, the idea that we just went over, polymers are a large unit of the macromolecule made up of many monomers, and monomers are small pieces of the polymer. They're the building blocks. So please take the time to look at this chart in the media album. I'm not going to go into it in detail right now, but this gets us to the point now where we can actually start talking about the biomolecules, what the sources or what the functions of them are, where we get them in a diet, and we'll go from there. So again, thank you for your time, and we will talk with you soon.